We were discussing the tumor antigens and had first dealt with tumor specific transplantation antigen, which is also called as rejection antigen. Then I have come to the second part, that is second type of antigen. A tumor may occur in a mice due to injection or treatment of that mice with a carcinogenic agent, and that tumor that grows in the mice is called as tumor plus, and that tumor cells, when you transplant into another mice or inject into another mice, which is a syngeneic mice, means has got genetic constitution same. These are all inbred mice. So you will see that the transplant is going to be accepted, and that mice also will express the tumor, which is tumor plus cells, but does not express tumor specific transplantation antigens. Therefore, the tumor does not get rejected. Tumor will be accepted. Later, this mice which has received the transplant should develop the immune response against that transplant, because it will recognize the antigens on the tumor, which is different from the normal cells, and starts building up an immune response against the tumor, and slowly the tumor will regress because of the CTL response and NK cell response. Let us see what happens. Now, those tumor cells of the mice which are being obtained from another mice have been in vitro mutagenized. They are treated for the second time with another mutagenic agent, and those tumor plus cells, after treatment with the mutagenic agent, now injected once again into syngeneic mice, the mutagenized clones. Here you will see, surprisingly, some mice will develop the tumors once again, and they will be exactly similar to the mice which had developed tumors when that tumor plus cell was injected into that mice or transplanted. And another clone of mice which was injected, syngeneic mice, will not develop the tumor. Okay, so that indicates that there is variation in the expression of genes, the mutagenized tumor cells. That means the mutations that occur in the cancer cells due to chemical insult or viral insult or radiation, whatever, it is not necessary that all cells that are exposed to the mutagen should mutate. Some cells will mutate, and some cells will show a different type of mutation, and that does not qualify those cells to continue as tumors. Okay, and possibly when you transplant that, because you injected that, those tumors which are developing in the mice are because of the new antigen that is expressed by the tumor. And some mice, when you inject, will not develop the tumors because they are expressing those transplantation rejection antigens. And when you analyze these tumor cells, it is observed that the mutant antigens are not always membrane bound. They are intracellular. Since they are intracellular, they are processed by the class 1 MHC. And when they are processed by the class 1 MHC and presented on class 1 MHC, they are going to be detected by CTL and NK cell in the mice beforehand when you inject them, and therefore they are not allowed to grow into tumors. So therefore, in vitro mutagenized tumor cells, some will fail to grow in syngeneic mice and some will grow in the syngeneic mice. Those which grow in the syngeneic mice are the ones which have developed new antigens because of the mutagenesis. Okay, and those which do not grow are the ones which are expressing now the transplantation rejection antigens, which were not there in the earlier stage. Because of the mutation, they are expressing the transplantation rejection antigen, due to which immune response in that mice will be very high. Therefore, tumor does not grow. So, one can understand from this the tumor cells' behavior. The tumor cells behave differently with the mutagenic agents, and the expression of antigens vary in the same line of tumors, in the same line of clones. And it is also observed in the person where the cancers are developing in solid tumors. When the solid tumor is analyzed, when the molecular analysis is done of the small solid tumor, it is observed that there is different population of cancer cells in that solid tumor. And each cancer cell is unique by itself. And that is why it becomes difficult to pinpoint and target a cancer cell treatment. And when you start the treatment, some cells will respond and some cells will not respond. And those cells which do not respond are the ones which will carry forward the tumor growth later, after the treatment is completed or once you terminate the treatment. That is how tumor antigens behave and tumor antigens are expressed, which gives some added advantage for the tumor cells for the growth. And that can be proved through immune response as well as by molecular identification of those antigens. Then we will go to the third type of antigen, that is tumor specific shared antigens. Now, shared means they are not only special to the tumors, they are also present in normal cells. But in normal cells, they are generally silent, very silent, because they are not expected to be expressed in the normal cells at that particular time when you have harvested the normal cell and analyzed them. And they might have played a very important role in the early embryogenesis. And plus, when the differentiation of the tissue took place and the cells of that particular tissue formed from the stem cells or from the embryonic cells, those antigens or those proteins or glycoproteins had played a major role in the early development of those cells to form that organ. Since they were formed early, and I had already told you that whatever the antigens that express early in the life of an individual, against those antigens, the individual develops tolerance. And that is called a self-tolerance mechanism. And I had already explained to you the experiment carried out by Bellingham and Peter Medawer, where early neonatal mice were exposed to the antigens. By injecting those antigens at the neonatal stage, they develop tolerance to that. Or embryonic stem cells, when they are mixed together, Beatrice experiment, there also they develop tolerance. Similarly, here also, the embryonic antigens or the antigens which had played a role in embryonic condition will be tolerant. And now, suddenly what happens is when the tumor grows of a particular tissue, these embryonic antigens are overexpressed. And when they are overexpressed, sometimes with some mutations in them, but in major part, they are almost similar to the normal antigen. And because of one or two amino acids different in them, it will be recognized as a strange antigen, and they are overexpressed by the tumor. And such shared antigens, which are there in normal cells and are present in the tumor cells also, but in the tumor cells, they are overexpressed and spontaneously expressed, will result in developing immune response against them because they are differing by the original antigen or the antigen that was expressed in the embryonic condition that differ from them by 1 or 2 amino acids. First antigen that was recognized is as MAGE, M-A-G-E. The full form of that is melanoma antigen, AGE for antigen. There are about 3 different melanoma antigens, MAGE1, MAGE2, MAGE3. And these antigens are predominantly expressed in melanoma. Melanoma is the skin cancer. And the person who develops melanoma on the skin develops hyperpigmentation in that area because the melanin producing cells have become cancerous. They have undergone transformation, due to which they produce excess amount of melanin pigment, and that area becomes black patch. Therefore, it is referred as melanoma. Large areas in the body will become black patched areas, indicating the person has developed melanoma. And on analysis, it was observed that melanoma expresses MAGE antigen. And then it was found that that antigen, which has got nothing to do in the breast cell cancer, is also expressed in breast cell cancer, breast carcinoma, expressed by placental trophoblasts. And placental trophoblasts is nothing but those are the cells that facilitate blastocyst implantation in the uterus and in the testicular germ cells. Melanoma antigen MAGE1 is expressed in breast cancer cells, placental trophoblasts, testicular germ cells. Now, when I say placental trophoblasts, they are normal cells. Testicular germ cells are normal cells. Breast cancer cells are abnormal. See now, the antigen that is expressed in melanoma is expressed not only in cancer, in normal cells. And that becomes difficult for the treatment targeting these antigens, using these antigens as target antigens by using monoclonal antibodies, because they are there on the normal cells also. And the cross reactivity will result in destruction of normal cells. So there are other antigens similar to the MAGE, that is RAGE. RAGE is supposed to be renal antigen. GAGE is supposed to be gonadal antigen. Like that, many such shared antigens have been identified, which are not only tumor specific, they are also present in normal cells. But in the tumor, they are overexpressed. That's the only difference between the tumor and the normal cells. And sometimes when you get the analysis done, you see that these antigens are differed by the original antigen by 1 or 2 amino acids. And the difference by 1 or 2 amino acids may not make them qualified enough to develop an antibody treatment against them, because they will not differ majorly from the self antigens. And there is a possibility that they will cross react with the self antigens the monoclonal antibody developed. It is similar to, remember, molecular mimicry. We had studied in autoimmune response molecular mimicry. In molecular mimicry, the antigens slightly differ from the normal antigens of the host cell. And that is the reason that tumor specific shared antigens are not used for treatment purpose, or not used for developing the monoclonal antibodies. They are there for the identification purpose. They can be identified, and one can say that this is the expression of the differentiation antigen, given some extra ability for those cells to grow into tumor. Then comes the fourth type of antigen. The fourth antigens are expressed by the proto-oncogenes. Proto-oncogenes. All of us carry these proto-oncogenes, but they are not cancerous genes. They are normal genes. And these normal genes, the proto-oncogenes, anytime can get mutated for various reasons, due to chemical carcinogenesis, viral carcinogenesis, radiation carcinogenesis, or due to chromosomal anomalies, chromosomal aberrations. and convert themselves from proto-oncogenes to cellular oncogenes. Cellular oncogenes. Proto-oncogenes are normal. Cellular oncogenes are abnormal. They are the mutant version of proto-oncogenes. When they are the mutant versions of the proto-oncogenes, the protein products produced by the cellular oncogenes will not be the same as the protein product produced by the proto-oncogenes. Since majority of them express those proteins and some belong to the membrane proteins as glycoproteins and proteins, and some of them act as growth factors, some of them act as growth factor receptors, some of them are signal transducing elements, and there are some intranuclear proteins which facilitate transcription. They are transcription factors that are the products of proto-oncogenes. And some are very, very high regulatory proteins. They're responsible for programmed cell death, that is apoptosis, and some of them are regulators of cell cycle, and they are called as tumor suppressor genes. So the proto-oncogenes, which perform different function, normal function in the cell, some of them belong to tumor suppressor category. They do not allow the tumor to occur because they keep the cell cycle under regulation. But on mutation, they will lose that ability of tumor suppression, or they may overexpress some certain proteins on the membrane of the cancer cells, and overexpression of those proteins on the cancer cells gives the malignant feature for the cancer cell. And sometimes some of them act as growth factors and produce that growth factor in excess, which will be facilitating cancer growth. And some are growth factor receptors, or maybe intracellular, the nuclear transcription factors or signal transducing factors. So there will be qualitative difference as well as quantitative difference in the proto-oncogenes when they move into cellular oncogenes. And that qualitative and quantitative alteration of the property of the cellular proto-oncogenes gives the additional ability for the normal cell to undergo transformation and develop into tumor. And some of them, the tumor suppressor genes, when they are not working, and one of the tumor suppressor gene is known for apoptosis, causing apoptosis of the cells. And when it mutates, the apoptosis of the cell does not take place. Even though the cell is abnormal, it does not die. It will remain immortal. And that is the characteristic that the cancer cell gains. So those proteins which are produced internally, which are there performing different functions, are presented on class 1 MHC by the cells when they are proto-oncogenic projects. Okay, and when they are proto-oncogenic products, they are considered as self antigens. They are considered as self antigens, so there is no immune response. But when they mutate and transform and they produce different types of proteins, they initiate T cell response, that is CTL and NK cell response. Therefore, initially, when the proto-oncogenes have undergone mutation, there will be tremendous amount of CTL and NK cell response against these tumor antigens. Okay, and these altered proteins are going to be expressed on class 1 MHC as the micropeptide. Now let us say that the tumor cell dies. If the tumor cell dies in the initial stage, it may be phagocytized by the antigen presenting cell, and macrophage plays a major role in scavenging of the dead cells. And that proto-oncogene product, which was mutated and expressed by the tumor, which has grown because of the mutation in proto-oncogene, now going to be processed by the antigen presenting cell because it has phagocytized that dead tumor cell. Then once it processes that antigen, it will present it on class 2 MHC. So there is possibility that mutant version of the proto-oncogene products may be presented on class 1 MHC, Generally, they are presented on class 1 MHC, but occasionally they will get presented on class 2 MHC by the antigen presenting cells. Once it presents on class 2 MHC, the T cell, that is T4 cells, are going to be kept active against the tumor to produce lot of cytokines which are responsible for the destruction of the tumor. Okay. Tumor gets targeted because of the presentation of antigen on class 1 and class 2 MHC. So the tumor antigens originating from mutation of the normal proto-oncogenes could also serve as antigens for the development of the immune response. And the T cell immune response is very strong against these antigens, which is recognized. Okay. Then we will go to the another part where the proto-oncogenes which are there in the cell may undergo mutation by the oncogenic viruses, and they are RNA and DNA tumor viruses. So RNA tumor viruses, which are called as retroviruses. I have given 3 examples here. There are many more. One of them, first discovered RNA tumor virus, is called as Roux sarcoma virus, named after the scientist Peyton Roux. Silent Peyton Roux. He had identified this sarcoma, that is muscle cancer, in chicken. They were developing tumors, and when he analyzed these tumors, he found that the tumors contained a RNA tumor virus, and that virus is named after him as Roux sarcoma virus. And that Roux sarcoma virus produces a protein called as SRC protein. SRC protein. Proteins are always represented by first letter capital. And the oncogenes, this protein is produced by an oncogene now. I will call it as an oncogene. Why? Because the proto-oncogene has undergone mutation because of the virus, and it is producing the SRC protein. Now, when I go back to the proto-oncogene that is there in our cells also produces SRC protein. SRC protein is found to be an intracellular domain of a receptor in our cell, normal cell. Say, for example, EGF receptor, epidermal growth factor receptor. So, epidermal growth factor receptor, which is present on the normal cell, facilitates the mitosis and growth of the normal cell when the receptor is bound by the epidermal growth factor. Okay. And this SRC domain, protein domain, is present in the intracellular region of the epidermal growth factor receptor, EGF receptor. Now, the virus, which is Roux sarcoma virus, if it infects the cell, it has got homology. The domain of its has homology to SRC domain of the EGF receptor. Because of the SRC domain of the EGF receptor, this virus, after the production of cDNA, because it is a retrovirus, from the RNA, it produces the cDNA. And that cDNA gets incorporated at the same site where the proto-oncogene for SRC protein was present. And now it will produce SRC protein belonging to Roux sarcoma virus. And the behavior of this SRC protein of the Roux sarcoma virus will be different from the SRC protein of the normal EGF receptor. And that gives the additional ability, because of the behavior of the SRC, the tumor gaining property for the cells. Similarly, there is another virus, avian myelocytomatosis virus, AME, avian myelocytomatosis virus. And this virus causes erythroblastosis in birds. Okay, red blood cell cancer, erythroblastosis. And it was observed that this virus carries a gene, that is MYC gene. The gene is always represented by small letters, MYC, MYC. Similarly, SRC, small letters. SRC, small letters represent gene. The gene product, that is protein, with the capital letter, beginning with the capital letter, MYC protein. And this MYC protein is responsible for cell proliferation. It facilitates cell proliferation. Once again, normal cells have c-MYC gene. C-MYC gene, cellular MYC gene, that is proto-oncogene, which regulates the cell cycle, which is very well balanced product produced and keeps the cell cycle under control. But on mutation, it will resemble exactly the avian myelocytomatosis virus and in nature, and it gives extra ability for the cell to undergo proliferation. It facilitates the cell to proliferate. Now, what am I trying to arrive at? The proteins that are produced, which are similar to the proteins of the cellular origin, that is proto-oncogene, due to the viral mutation. In the earlier example, I had mentioned that viruses can also bring out mutagenesis of the proto-oncogene. Now here I am talking about RNA virus causing that mutation, and the protein produced by that RNA virus resembles the protein of the cellular proto-oncogene, almost in all respects, except that few of the amino acids are different. And because of that, those amino acid differences make that protein different, gives the extra ability for the cells to undergo proliferation and develop into tumors. And these proteins will be presented on class 1 MHC. And when they are presented on class 1 MHC, they are abnormal proteins. They are going to be recognized by the CTL and NK cells, and the immune response is mounted against the RNA initiated cancers, RNA tumor virus initiated cancers. Kirsten sarcoma virus produces a protein called as RAS protein. Okay, like that, there are many examples of the RNA viruses which are responsible for causing the tumors in rat, in mice, in birds, as well as there are similar proteins expressed in human tumors, human cancers. Okay, human cancers. And these proteins are responsible for the excessive proliferation of the cancer cells. And there is immune response recognized against these proteins in the cancer cells. Then we'll take up the example, that is DNA tumor viruses. Okay, DNA tumor viruses, similar to RNA tumor viruses, there are DNA tumor viruses which frequently infect the human beings. And there are known cancers in the human beings of DNA tumor viruses. And very strange cancers are there in human beings of RNA viruses. Okay. And one example I have not given you, the RNA virus, that is HIV virus, which is an RNA virus, is known to cause Kaposi sarcoma in human beings. And Kaposi sarcoma is nothing but small tumors which emerge on the skin, and they are the dermal cell cancers. Large number of tumors appear on the skin as bulgings in AIDS patients, which are formed because of the HIV virus, which is an RNA virus. Then when you talk about DNA virus, there is a virus called as Epstein-Barr virus, named after the two scientists Epstein and Barr. And Epstein and Barr had discovered this virus. And it is very common in the persons who develop Burkitt's lymphoma. And the Burkitt's lymphoma is characterized by swelling in the lower jaw of the person. And it is the lymph node of the lower jaw of the person undergoes transformation. And there you will see the population of these Epstein-Barr virus will be in excess in that transformed cells. And when you extract these viruses and transfect a fresh population of lymphocytes, Epstein-Barr virus extracted from Burkitt's lymphoma patients from their lymphoma and infected to the fresh lymphocytes, which are not yet cancerous, which are normal lymphocytes. Those lymphocytes undergo transformation. Those lymphocytes become cancerous, which indicates that Epstein-Barr virus has the ability to bring about transformation of cells, mutation of cells to develop into tumors. And Epstein-Barr virus protein, I had given the example One of the protein of that is DNA polymerase, and that DNA polymerase mimics, remember, it is capable of mimicking the myelin basic protein, and the person suffers from the the muscular sclerosis, right? So the person suffers from sclerosis and multiple sclerosis, which is the cause of the degeneration of the nerves. And that Epstein-Barr virus is a potent tumorigenic virus. The protein produced by that is also different. Similarly, hepatitis B and hepatitis C virus both are known to cause hepatocellular carcinoma, liver cancer. Then there is a virus called as Show papilloma virus, which is called as SPV virus, causes the skin rash initially, and that skin rash transforms into a skin cancer, epithelial cell cancer of the skin, Show papilloma virus. And you have heard about human papilloma virus, HPV, which causes the uterine cervical cancer. Cervical refers to here uterus, not the cervix cancer. And the cervix cancer is also referred as cervical cancer, and that is the uterine cervix which develops the cancer because of the human papilloma virus. Now, all these DNA tumor viruses and RNA tumor viruses are opportunistic viruses which infects the cell, and they have what is the commonness with the cell is the homologous sequences in their DNA, that is in the DNA tumor virus, or the RNA which synthesizes the complementary strand of the DNA. Therefore, that complementary strand of DNA can recombine with the normal cell genome at the same site where that particular gene is supposed to be active. And that gene is active as well as the gene is under regulation. But on the integration of the viral genome into the genome of the host, which is called as insertional mutagenesis, that causes the new proteins to be produced in an unregulated manner, or the proteins with difference in amino acids, which helps in recognition of the tumor because these proteins are expressed and then they are processed and presented on class 1 MHC, which targets these tumor cells by the CTL and NK cell response. So the virally transformed cells of DNA or RNA virus are attacked by the immune response because they express strange antigens. Then comes the another group of antigens which are expressed by spontaneous mutation. Okay, those are spontaneous mutations which take place of unknown reason. What reason that causes those mutations? Nobody knows. And these mutations give extra ability for the tumor cells to develop as full-blown cancer. One of them is cyclin-dependent kinases. There are CDKs, cyclin-dependent kinases. I think you have done it in cell and developmental biology. These cyclin-dependent kinases regulate the cell cycle. They appear at different stages of the cell cycle. Some cyclin-dependent kinases appear at G0 phase, some appear at G1 phase, and some in between G0 and G1, and then some will appear at G2, and some will appear at M phase, and therefore the cell cycle is completed in a regulated manner. But imagine these cyclin-dependent kinases, which are under regulatory control of another protein, okay, and that too, that protein is a tumor suppressor protein. That protein regulates the cell cycle, does not allow the cell cycle to undergo haywire. If the cell cycle undergoes haywire, then only the abnormal growth of cells will take place, means large number of cells will be formed. So the tumor cells have adopted a mechanism, the cyclin dependent kinase, which is capable of responding to a regulatory protein, cell cycle regulatory protein, has been made unresponsive by mutation, so that regulatory protein cannot bring about the control on the cell cycle. Therefore, the mutated version of cyclin dependent kinases will help the cells to undergo proliferation in an uncontrolled manner. The mutant version of CDKs help the cell to undergo in an uncontrolled manner, therefore the tumor growth takes place. They will not respond to tumor suppressor proteins. That is one added advantage to the tumor cells. Now, why do they undergo such change? Is because of the spontaneous mutations in them. The spontaneous mutation means it is a different protein, and different protein means it has to be recognized on class 1 MHC. Similarly, beta-catenin. Beta-catenin is a protein which is present in all stem cells. Wherever the stem cells are there in our body, which are emergency cells, on demand, they undergo division and provide the new batch of cells. And I told you stem cell characteristic is very strange that in the two daughter cells, one of them will retain the stem cell characteristic and another cell will not retain the stem cell characteristic. It undergoes differentiation to give rise to the particular cell that is in demand of a particular tissue. And this will happen as a tissue injury. Some cells in a particular tissue are lost. When the some cells are lost, the stem cells contribute to replace those lost cells. Okay, and when they are contributing, the stem cells have to still retain the stem cell characteristic because at any time they should be ready to provide the new batch of cells. To retain that stem cell characteristic, beta-catenin facilitates. And stem cells behave like undifferentiated mass of cells. And that is one of the important characteristics for the cancer cell. And cancer cells behave like undifferentiated mass of cells. They mimic all embryonic conditions of the cells. So therefore, they keep beta-catenin active. The beta-catenin in normal cell does not stay beyond 20 minutes. 20 minutes is the half-life of the beta-catenin in normal cell. And within 20 minutes, it has to finish its work and get destroyed by the ubiquitination. But the mutant version of beta-catenin does not undergo ubiquitination and it does not perish. It lives in the cell for more than 20 minutes. Because it lives in the cell for more than 20 minutes, it gives that additional ability for the cancer cell to retain stem cell characteristics so that they can continue to proliferate as stem cells, retaining stem cell characteristics, but not differentiate. So therefore, the differentiated mass of cells which have undergone transformation gain the ability of embryonic ability, that is stem cell characteristic, because of the beta-catenin which is mutated. And that also is recognized by the immune cells as a strange protein. Then there is a protein called as caspase, which is responsible for the apoptosis. Cancer cells in the beginning form, many cells die because there are strange mutations which are not good for the cells. So those cells die. But later, the cancer cell adapts in such a way and it evolves in such a way that it will tolerate those mutations and then it prevents the apoptosis. It doesn't allow the cancer cell to undergo apoptosis. So that is because it will mutate that caspase gene, and that caspase protein produced is not capable of causing the apoptosis. Similarly, the chimeric proteins means you don't see them in the normal cells. And these are called as fusion proteins. Generally, the normal cells, whenever one particular gene acts, that gene product will be a particular protein. Now here you see two genes will be brought together, like say for example, the gene that is the product is BCR and ABL. Both these are genes, BCR in small letters, ABL in small letters. Both these are small genes present in two different chromosomes, performing normal functions in the cell. And when these two genes are brought together, okay, because of the translocation of the chromosome and exchange of the segment of the chromosome, you will start getting a fusion protein. Means BCR protein will be fused with ABL protein and a new protein will be produced, and that fusion protein is responsible for giving proliferative ability for the cells. And that fusion protein will be recognized by the CTLs as a new protein, and the immune response builds up against that. Okay. Then MYC, I have already explained to you. There are MYC proteins which are produced as mutant varieties, and those mutant proteins do not respond to any protein which controls the cell cycle. And that mutant protein also is recognized by the CTL. So therefore, the 6 different categories of different types of antigens expressed by the tumor cells, all of them are capable of inducing the immune response through T cells. And why it is T cells? Why? Because they are presented on class 1 MSC. Okay, CTL and NK cell response is very, very good in tumors. In later part of the lecture, you will come to know that almost all solid tumors will be always infiltrated by the cytotoxic T cells as well as NK cells. And large number of these cells will be there in the solid tumor, and they are causing the death of the tumor. But one fine day will come that these cells also will be helpless, because tumor cells will have a resistance ability of killing mechanism of these two cells, and they will develop resistance to killing, and they don't die. Therefore, the cancer succeeds by suppressing the immune response or evading the immune response. That we'll be talking about that in the later part of the lecture.